Okay. Lesson 5. Bacteria and their effects on humans. Bacteria are classified according to their effect on humans in the following categories. Harmful or disease-causing. Bacteria are known as pathogenic bacteria or pathogens and cause various diseases in humans, animals, and plants. A person cannot see, smell, or taste pathogenic microorganisms in food, but can become very ill from them. Food contaminated with pathogenic bacteria has no change in taste. I repeat, food contaminated with pathogenic bacteria has no change in taste, smell, or appearance, and is difficult to identify. Victims of foodborne illness usually do not detect any change in the taste of the food that sickened them. Undesirable bacteria, which cause decomposition of food, are often referred to as putrefying. I repeat, putrefying bacteria. That means undesirable. These bacteria accelerate the decay and decomposition of food. Spoiled food is easy to identify by changes in the color, taste, odor, and texture of the food. However, spoiled food is not a cause of foodborne illness. Because of obvious changes in smell and color, spoiled food rarely ends up being eaten. Next, beneficial bacteria. Beneficial bacteria are used in the production of various foods, including cultured milk, yogurt, cheese, and sauerkraut. Last, benign bacteria. Benign bacteria are neither helpful nor harmful to humans. Of the hundreds of thousands of strains of bacteria, most fall in this category. So most categories are benign. Most bacteria are benign. Next. In order to understand food sanitation practices, it is necessary to know a few facts about the microorganisms that cause food spoilage and foodborne disease. Bacteria. Bacteria, commonly called germs, germs, are extremely small plant-like organisms that can be only seen with a microscope. So you can only see bacteria with a microscope. 1 million could fit on the head of a pin. Like any living thing, bacteria require food, moisture, and the proper temperature for growth. Bacteria are found everywhere on the earth, in air and water. The soil abounds with bacteria that grow on dead organic matter. The main objective in protecting public health, is to control and destroy pathogenic, I repeat, pathogenic, disease-causing bacteria and those that cause food spoilage. Proper food handling and storage can keep these organisms to a, and that says at the bottom, and you can't see it, but I would say to a minimum. Spores. Some bacteria are able to protect themselves under adverse conditions by forming a protective shell or wall around themselves. In this form, they remain dormant while awaiting the right conditions for them to grow again. Dang, so they put themselves inside of a shell until the time is right. That's what they do. They build a wall around themselves until the time is right to grow. During this dormant stage, they are called spores. Bacterial spores can be likened to the seeds of a plant that are also resistant to adverse conditions and only grow when conditions become favorable. So they could change themselves into a spore, protect themselves in a spore. until the conditions right for them to grow. During the spore stage, bacteria do not reproduce or multiply, but with proper warmth, moisture, and air, they resume their normal vegetative stage and growth. Since spores can withstand rigorous conditions, they are difficult to destroy. Fortunately, only relatively few pathogens, for example, tetanus, anthrax, and botulism, are spore-forming bacteria. Go to the next. Bacteria reproduction. Bacteria reproduce by splitting in two. This is called binary, I repeat, binary fission. Fi, fission, binary fission. Therefore, their numbers are always doubling. One bacterium generates two, and each of these generates two more, resulting in a total of 4. The 4 then become 8 and so on. Dang, them prickers grow, multiply. Dang, it just keeps splitting and splitting. Rapid bacterial multiplication compromises food safety. This happens under certain conditions, including moisture, warmth, nutrients, and time. That's why you need to know you keep it in the right temperature, the danger zone. Under ideal conditions, one organism can become 2 in as little as 20 to 30 minutes. The table below shows bacterial multiplication in a food that initially contained 1,000 organisms. Here's the number charts. 2,000, 30 minutes later. 4,000, 1 hour later. So 8,000, 1.5 hours later. So on and so on and so on. So every 20 to 30 minutes, it's splitting in 2. Oh, look at that growth curve. Jesus. That is nasty. That's bacteria and affecting human body. Okay, this ideal rapid growth is called the log phase, when it keeps splitting out. All bacteria will reach this rapid part of their growth under proper conditions. Bacteria begin their growth cycle by adjusting to any new environment or condition by being in a resting lag phase. Stationary and death phases occur with the depletion of available nutrients and the production of their waste products. Please see the figure for bacterial growth curve. Okay, if you strip some nutrients from them or what they like to feed off of and put what they trying to get rid of, you kill them. That's the death phase. Lag, log when they begin to split up. Stationary when they stop dividing, and death phase when you strip the nutrients and get them put in with their waste. They can become a waste, they start to die.